Okay, so the next topic we're going to talk about is the muscular system. Now, when we talk about the muscular system, is a system that composed of many organs, right? I know you don't think of the muscle group as, say, um, you probably not think about this, but this one right here, the bicep brachii, is an organ. Okay, the tricep brachii is an organ. All the muscle that you guys start learning in week 2 in the lab, all of them is an organ, right? And their function um, and organs. The reason why it's called organs because every organ has to have more than one tissue. Organ has more than one tissue, right? And what are the tissue? Every tissue has epithelial. I mean, every muscle has an epithelium. That's one tissue. It has connective tissue like blood flow in them. It has neural tissue because skeletal muscle won't contract until the motor neuron innervates it. But all of the organs within the skeletal or sometimes um, the smooth muscles and cardiac muscle function, right? This muscular system function, if you want to say, uh, function is to contract. That's it. Okay. As a skeletal muscle function, certainly contraction might be important for movement, right? Contraction might generate heat in the case of shivering. Contractions, particularly some of the uh, sphincter, like a circular muscle, to regulate the movement, if you will, how the chyme from the stomach empty into the duodenum is regulated by the pyloric sphincter, for example, or even your anus sphincter. When it's contract, it allows you to hold the fecal material, and the relaxation of the external anus sphincter uh, allows the fecal material to pass through. Uh, Muscle contraction, particularly in the face, it may be involved in facial expression, for example. So those are all of the functions of the muscular system. But keep in mind, it's an organ. Now, uh, it has more than one tissue. What are the four tissues? Four tissues, keep in mind, are epithelium, right? Connective, neural tissue, neural, nervous, and um, uh, connective tissue, epithelium, neural tissue, and muscle tissue. Okay? So keep in mind is that muscle tissue is different from muscular organ. Okay? Muscular organ has muscle tissue. Right? What are the muscle tissue? Well, there are three kinds of muscle tissue. Muscle tissues are is a skeletal muscle, skeletal muscle is cardiac muscle and smooth muscle. Right? Okay, so let's start the lecture with these muscle tissue. Now here we're talking about muscle tissue, and the three muscle tissue are cardiac, smooth, and skeletal. And again, anatomy and physiology is same thing as structure and function. These three muscle tissue can be distinguished from one another based on what they look like, anatomy, right? What I mean by that is that of these three muscle tissue, the skeletal muscle and cardiac muscle have striation. Okay, that's the key. These two have striation, whereas smooth muscle do not. And by striation, we mean that there is patterns of dark light, dark light. Okay, so striation is a pattern of dark light, etc. Okay, and so if you look at the muscle, I'm just going to draw a cartoon of it. There's this dark here region. There's a light. There's a dark. There's a light. There's a dark. You can see here. There's a dark, light, dark, light, dark. This is striation, and you can see in the skeletal muscle and cardiac muscle has striation, whereas smooth muscle do not. But you can further distinguish between skeletal muscle and cardiac muscle, and that is the cardiac muscle branch. It will be like this, whereas the skeletal muscle do not branch. That's one key thing. And cardiac muscle typically communicate with each other via this gap junction. And you see a lot of that gap junction perhaps over here, like that. Okay, this is known as intercalating disc. So the presence of intercalating disc will tell you that it's cardiac muscle. Okay. Furthermore, if you look at the nucleus in the cardiac myocyte, they tend to be in the center, right here. Now, in the case of skeletal muscle, they have striation, but this striation is actually due to the highly arrangements of actins and myosin. We're going to look into detail what that is. But because there's a highly arrangement, they tend to push the nucleus to the side. Okay, so in the skeletal muscle, the nucleus tends to be on the periphery that you see here, not in the center. Okay. That's anatomical descriptions of these three different muscle tissue. Functionally, they also behave differently, and that is skeletal muscle is the only type of muscle tissue that you can tell it when to contract, right? I can tell the muscle in my hand to contract now or to relax, right? Or to contract now, the bicep brachii or the tricep brachii contract to extend. So we call that is voluntary, okay, under conscious control. So the skeletal muscle is voluntary. Whereas the cardiac muscle, myocyte, or the smooth muscle, it is control, it is contraction where you can't control it. So we'll call that involuntary. Okay, so this is involuntary, and this is involuntary. So that's not under conscious control and not unconscious control. That's functional characteristic called involuntary. And if you want to go further and say that involuntary, that particularly smooth muscle, right, it only contracts when it's responding to the local environment, right? Whereas the skeletal muscle is contract because of motor neuron innervates it, right? So skeletal muscle require a motor neuron to stimulate it. The cardiac myocyte can contract by itself, okay? So it's known as, it undergo a spontaneous depolymerization at a particular place known as the sinoatrial node, or known as the SA node. As such, it sets the pace of the heart, also known as the pacemaker. So the cardiac myocyte is self-excitatory, if you will. Okay, this right here is self-excitatory. Okay, and it does it as a regular at the regular interval. So we call that there's a rhythmic auto. We call that auto rhythm. Myth, rhythm. Okay, so rhythm, rhythmicity. Okay, autorhythmicity, autorhythmicity. Okay, whereas the smooth muscle responding to local environment. We can, there's a couple of examples. One example will be the precapillary sphincter, and we can come back to this later. The precapillary sphincter is our smooth muscle that regulate how much blood can go into the capillary network. Uh, if there is sufficient oxygen in the area, so there's no reason why you want more oxygen. So you have the precapillary sphincter constrict, and then so the blood is then doesn't need to go into that capillary network. If the oxygen level is low, the precapillary sphincter open up and the blood can pull into the capillary network, right? And so the local environment regulates that sphincter. Similarly, in 301, you might see that how if you swallow a bolus of food that gets down into the esophagus, the extensions of the esophagus, the smooth muscle, causes to contract and relax, contraction and relax. This is known as peristalsis, and doing so will propel the food down. This is a similar thing is that if you have a tube of sock and you insert a tennis ball in there and you just squeeze in that one area above, superior to the tennis ball, let it go. Squeeze, let it go. Squeeze, let it go. And you can see pretty soon the tennis ball will move down the tube sock. And that is because the bolus of food, or the tennis ball in this case, stretch the local environment, the smooth muscle, then it causes to contract. Okay. Okay, let's move on then. Um, so as a muscle organ, you can see here, organs of the muscular system has muscle tissue is one of these, but it also have other tissue, nervous tissue, blood is part of the connective tissue, as well as many other connective tissue. Um, I know you guys will do a lot of virtual lab now, but if we meet in person and you do dissecting of cat muscle, for example, you probably won't see the muscle unless you isolate it. And by isolated, you might have to remove the loose areolar connective tissue, which is basically kind of like spider web, if you will. That's a connective tissue that you have to remove. Um, Now, a lot of these muscle tends to exert cells on bone as part of the tendon, so that if the muscle move or contract, the tendon attaches to bone, causes that bones to move along with it. But in this case, what you see these um, uh, connective tissue known as fascia, okay, which is basically connective tissue that cover the muscle. That fascia can serve as an anchor for the other muscle, uh, and it forms what we consider as the aponeurosis. And you have one here called the epicranial aponeurosis or the abdominal aponeurosis. And these, in the abdominal, you have the linea alba, may serve as an anchorage for the rectus abdominis that may be involved in flexion of uh, the torso. Uh, and if you want to do a lot of crunch, your rectus abdominis might hypertrophy. Then you can see a nice, beautiful six pack in that person, I guess. Uh, the two external muscles here called external abdominal oblique start from the thorax that comes down, and the one that inside that goes from the iliac crest that goes up. So you get this mismatch, right? So the one that goes down. So you usually put your hands down like this. Okay, that's your that external abdominis oblique, and this one that goes up we call internal abdominis oblique. Um, Diagonal, so hence the name. Um, every muscle tends to have two insertion sites, the origins and the insertion. I'm not going to talk about too much other than to say that if you want to understand um, how uh, what is the functions of that muscle, you have to know two uh, attachment site. One is known as the origin, which is the immovable uh, attachment, and then you have the insertion, which is the movable attachment. That's all I'm going to say. You can just say, well, the bicep brachii is example, right? So the word bicep means two. It has two heads. The bicep brachii origin is up here as part of the coracoid process of the scapula. Okay, and it runs across and it may insert on the radius, for example. And when the bicep brachii contract, you can see this one doesn't move, but this insertion here, the insertion move, and it causes the arm to go up like this. So we'll call this flexion. Likewise, the tricep brachii, when it contracts, it will extend the arm. Okay, so flexion and extension. That's all. That's the origins and that's the insertion. Okay, so connective tissue covering the muscle is very similar to how, if you are my 301 student, I want you to know that a nerve is a bundle of axon. So if you said this is an axon here that is coming out or going in, the connective tissue that is surrounding this individual axon is known as endoneurium, right? And then you have connective tissue that help forms as a group of axon like this into a fascicle. This is known as perineurium. Perineurium. And then a connective tissue that is surrounding the outermost is known as the epineurium. Epineurium. Okay, and the one innermost is known as endoneurium. Endo. Now, the reason why I bring this up is because in a similar arrangement, right? So you learn by process known as association. So if you know how a nerve is organized, you can now know how the group of muscle is organized. So now let's say that if this is a muscle, you see here, so the innermost connective tissue that's surrounding the individual muscle is now known as endomysium. See, endomysium. And you also have a group of muscle that forms into this structure here, with this structure known as the fascicle. Okay? And the word fascicle means a rope-like structure. Rope-like structure. So that if you ever see a rope, you probably think of a like the three um, twine that sort of like twist each other into a rope. Okay, so think of each of those twine is a fascicle. So in this case, you have one fascicle, one fascicle here is another fascicle. So the connective tissue that forms the fascicle here, right, surrounding the muscle is known as perimysium. Okay. Perimysium. And the outermost connective tissue surrounding the muscle is known as the epimysium. Okay. Epimysium, which is the outermost here. All right. So that's what the word epimysium means. It's the outermost connective tissue here. And then within the epimysium, you have the perimysium that forms as the fascicle, and the connective tissue in around surrounding the individual muscle fiber is known as endomysium. Once you get into the muscle fiber, right, the muscle cell, also known as a muscle fiber, by the way, you have two uh, filament myofibril, if you will. Okay, the thick one is known as the myosin, and the thin one is known as actin. Okay, this is the myosin. Thick one is myosin, and the thin one is actin. Okay. All right, so let me erase this, and you can see here that tendon connects muscle to bone. The outermost is epimysium, and you can see how you have a group of them that forms as the fascicle. That connective tissue is known as perimysium, and within the perimysium, you have an individual muscle fibers. Connective tissue would be the endomysium. Endomysium. Uh, all right, so here's some more terminology. Sarcolema is the um, is the instead of calling this the plasma membrane is sarcolemma. The word sarco means flesh. Okay? The um, the inside any cell is considered cytoplasm, but inside the muscle cell is known as sarcoplasm. Okay? There's this network of a network of tubule, if you will, um, in that in a regular cell we call that sarcoplasmic reticulum. In this case, sorry, it's known as endoplasmic reticulum, but in this case it's known as sarcoplasmic reticulum. And one of the functions of the sarcoplasmic reticulum, keep this in mind, is to store calcium. Is to store function is to store calcium. That's the sarcoplasmic reticulum. The transverse T-tubule is the invaginations of the sarcolemma. Okay, so let's say here's a sarcolemma. Now, if you started to invade like this, then it's a T-tubule. See how it looks like a T-tubule? T-tubule, like a T. And next to that T-tubule is the network known as sarcoplasmic reticulum. This sarcoplasmic reticulum in the area next to the T-tubule might be enlarged, right? Like you see here. This is known as the sustainae of the sarcoplasmic reticulum. So the T tubule and the two sustainae of the sarcoplasmic reticulum will forms the triad. You can see that triad. And all of this is the arrangements of actin and myosin. And in the future, if I ask you what is the functional unit of the skeletal muscle, it's known as the sarcomere. The sarcomere is a functional unit of the skeletal muscle. So if you want to understand how does the skeletal muscle work, you have to look at the anatomy of the sarcomere. Okay, anatomy of the sarcomere. Okay, so I can continue this um, next week, but before we leave today, I wanted to write. I have to say maybe what is the transverse? That's the T tubule. That's this right here. Is the invaginations of the sarcolemma. Okay. Let me share with you. I'll do 10 minutes and draw. Okay, the T tubule, which is this right here in the middle, is the invagination. So here's the sarcolemma. By invagination mean that to come in, invagination. There you go. Now it looks like a T. Hence the name, the T tubule. T tubule, transverse tubule or T tubule is the invaginations of sarcolemma. All right, so I'm going to, for the 10 minutes or so, I'm going to use these eraser board, if you will, blank sheet. All right, so here is the sarcolemma. Now, if you have the sarcolemma invagination, it comes down like this. This is the T tubule. Sorry, that's a T tubule. And next to the T tubule is part of the sarcoplasmic reticulum, but it flattened out, right? So this is known as the cisternae of the sarcoplasmic reticulum. And here's another one too. And then the network, the cisternae of the sarcoplasmic reticulum. Keep in mind that the functions of the sarcoplasmic reticulum is to store calcium. All right, so I'll just tell you the story now. If the membrane is depolarized, right, which means allows the sodium to enter. So most of the sarcolemma is pretty much like every muscle, every cells, and that is there's a negative membrane potential inside. Okay, now if you open up the sodium channel, sodium will rush in. So it's go from negative to positive. So we call that depolymerization. Okay, depolarization. And that depolarization will travel down the sarcolemma, right? It goes down. And in fact, it's go down to the T tubule as well. And when it goes down to T tubule, it will talk to the sarcoplasmic reticulum to release calcium. And so by causes the sodium to depolarize, enter here, your end result is that calcium exit the sarcoplasmic reticulum. Okay, but that's physiology. Let's do a little bit anatomy, and I'll explain to you why you might have this patterns of dark light and dark light. Okay, so let's say this is a sarcolemma and a sarcolemma. Okay, now the myofilament that you see inside the skeletal muscle have these filament highly organized. This is known as myosin. Okay, so what I'm drawing here is myosin and myosin. And staggers between the myosin is this one. It's called actin. Okay, so you have your myosin, your myosin, and your myosin. And here's your actin, and so forth. Okay, I'm not going to draw it, but you can draw the rest of it. Here's the idea. And by the way, the myosin is kind of like if you ever guys play golf, uh, like a 2 irons or 9 iron, you have a shaft, and then you have the head that looks like that. Okay, so when you say the myosin here, is that there is a whole bunch of myosin grouped together, so it looks like that. But it only occur at the end. So this right here we call the head that's sticking out like that. That's the myosin. Okay, the myosin's head. It is considered a catalytic domain, if you will. It's the area that bind that use up the ATP and allows us the myosins to stick to actin. So there's a physical binding to this between myosins and actin. But that's physiology. Before that, I wanted to show you something else. Um, Now, I don't know if you guys remember when you were little. Like I remember in my 6th grade, you make a dye tie T-shirt, and basically you take a cotton T-shirt and you wrap it around with the rubber band and you soak it in a red color, for example, and then you take it out and you remove the rubber band and you see this pattern, right? Now, the idea is cotton is protein, and you can certainly stains it. Protein can take and stain. So if you take the 5 micron section of the skeletal muscle and you stain it, right? You stain it with a special stain, and you will see there are 2 areas. If there's an area that has a lot of proteins, it's going to stain darker, right? And so if you take this tissue and you stain it, and you can pretty much divide this so that you can see that this region right here and this region here, this is one region, if you will, and then the region. So because of this, the area that has a lot of proteins in this, okay, this is known as the A band, and this is known as the I band. So the striation is brought about is because you see the presence of A, I, and AI, for example. Okay, now if you don't remember which band is dark or which band is light, think of this. How do you spell dark? Is DARK. How do you spell light? Is LIGHT. Okay, so you see that within the A band, there's 2 filaments, right? There's myosin and this one right here, actin. So um, within the A band, you see actins and myosin. Now the question is, what kind of myofilament do you see within the I band? Is it actins and myosin or both, or is it actin alone, myosins alone, or both? What do you think? The I band is composed of
Acting.
great, only actin. All right. Now the histologist or other anatomist doesn't just give you a name of A and an I randomly. In fact, there is a pattern. The word I stands for isotropic. Okay, isotropic, and the word A stands for anisotropic. So what that means is isotropic means same, whereas anisotropic means not the same. So in fact, what you see is that if you look at the I band, it's consistently the same because it's only have uh, actin. But within the A band, it's not the same. It's because if you look closely, there is a region here. You see that? There's a region here that has only myosin. You see that? It's supposed to come out here. And you can see this regions right here that have um, myosin, right? And so in the A band, it's not the same because there's a region here is myosin, whereas the edge here is actin. So myosins and actin. So this area will be darker than this. Okay, because this is a little lighter, we call it just the eighth zone. Eighth zone is because uh, H stands for Hela. In Greek, it means light. Okay, Hela is lighter. And so in the future, if I say, okay, what is the eighth zone? Okay, the eighth zone will have what kind of filament? Eighth zone will have what kind of filament? Does anybody know? The eighth zone? Nobody know what the eighth zone will have? Okay. The eighth zone could only have myosin. Good. Now, in the middle of the actin, there is a Z line here. Z line. This is a Z line in the middle of actin, the Z line. Right? Z line. So in fact, one could describe that the distance between the two Z lines is a sarcomere. A distance between two Z line is a sarcomere. Right? Remember talking about that. Sarco is flesh, mere is unit length, like in meter. Okay. And in this, the Z line is that we're in the middle of the actin, it has tintin and stuff like that. Okay. But another way of thinking of sarcomere and how it's related to contraction is defined as, remember, one of the functions of the muscle is contraction. Contraction in this case, you can say, is defined as contraction is the shortening of the sarcomere. And so the idea here is that in contraction, these two Z line, in fact, will come closer. As you see that when it comes closer, uh, the later on or next week we will see that the contraction is due to this, we'll call it the sliding filament model, and that is actins and myosin slide past each other. If they slide past each other so that they're doing contraction, there's two areas that get shorter, the H zone and the I band. Okay, if you can see that it's contractions of the myosin will come closer, the edge of the myosin will come closer, so the eye band will get shorter. And this dent right here, it's going to be smaller. So then that's my questions in the future. Doing muscle contraction, which area gets shorter? The H and the I. Okay, so keep that in mind. So I'm going to end it there. Stop the